All right, then welcome everyone again. My name is Lynn. I was part of the 2019 iGEM project with Alexander and some other people, more specifically these other people. So we were a quite small team with 4 students. So we were Alexandra, me, Roy, and Alexander. And we got together in November or December of 2018, started theoretically working on our project and did most of the lab work over summer and up until October actually, because we wanted to get more results. So that was also like the scope of our timeline. And there we realized that it's very good to get the iGEM team together a bit earlier, just because we got into a bit of stress. Might have been because it's just for students. Yes. So as you know, synthetic biology in iGEM, we usually try to have a project where we address a problem of the world with synthetic biology. And we wanted to address the problem of bacteria who can produce things, but don't do them in the way that we want to. So bacteria can be very useful for producing things. And my example that I wanted to bring, Alexander already brought. So for example, insulin was one of the compounds that you can produce with bacteria, but other compounds are more difficult to be produced in bacteria. And some of the limitations can be that maybe the reaction is harmful to the bacteria, maybe it's just not efficient enough because the reagents are spread all over the bacteria and you want them in one place for the reaction to be efficient. Or maybe you have a complex or multi-step reaction and it just does not work in the bacteria. So one way to solve for these limitations is by compartmentalizing the cell, because this way you can really get all the things that you want to interact with in one part of a cell or in a bacteria. There's different ways to do this. One idea would be to have lipid droplets, and within the lipid droplets you can have the things that you want. We tried a different approach, and to do that we also did something that's very typical for synthetic biology, namely to look at nature, see how they solve a problem, and try to simplify it and engineer it ourselves. So the nature thing that we looked at was a phage-bacteria interaction. Phages are just viruses that infect bacteria. So let me quickly talk you through the phage-bacteria interaction that we looked at. Now, is this visible? No. Can I? Okay. Oops. Perfect. Cool. New technology. So this here is a bacteria, and here we have the phage, which is currently infecting that bacteria. So by infecting, what it does is it injects its own DNA into the bacteria, and immediately after it has done that, a protein shell forms around the viral DNA, and this is functionally a compartment. Over the course of some minutes or hours, this compartment grows in size because the DNA inside gets replicated. And once there is enough replication, on the border of this compartment, the new variants are assembled. And once all the variants are finally assembled, the cell lyses, dies, and releases all the variants. This is the phage-bacteria interaction that we looked at. And now our goal was to look at how this compartment is built and see if we could use this as a way to compartmentalize a cell. So to be able to build this compartment in this bacteria without the infection, or maybe even in other bacteria or other cells, we first need to find out what this compartment is made of. So we wanted to look at some genes of this phage and see which genes could be essential for this compartment. This was made a bit harder because the phage that we were working with was actually a jumbo bacteriophage, which had more than 400 genes, which was fun. So we first had to identify which genes would be the interesting ones. So we looked at expression patterns and which genes are expressed in the very beginning of the infection, and selected 10 of these to do our further experiments with. What we then did with these genes is we GFP tagged the proteins that they coded for. so that we could look at these proteins in the bacteria and see what they do, especially when infection happens, because that way we can kind of infer the function. So we then did a lot of microscopy, which I really liked, not at that time point. And we tried to see what happens in the bacteria. So what do we actually expect when we see the microscopy images? If we have a bacteria that is not infected, but it expresses that GFP protein of interest, the DNA is supposed to be just spread out all over the cell, because what I did not mention earlier was in this interaction, actually, at this step here, the bacterial DNA gets degraded by the virus, because the virus has no interest in the bacteria producing whatever the bacteria would like to produce. So it starts cutting up all the bacterial DNA, so that afterwards here, you would see the DNA as a kind of blob, because all the bacterial DNA is destroyed, while the viral DNA is still intact and still visible. And here you can actually also see where the compartment is localized, because all the DNA that survives is the DNA in the compartment. For the GFP, if we don't have an infection, we assume that a viral protein is not doing anything because the virus is not there. So we expect the protein of interest to also be just spread out over the cell. If we have an infection, however, we can see two things. Either the protein of interest is not actually involved with building the compartment in the beginning, and it's used for something completely different, then we, in the beginning of the infection, still think that it's going to be spread out over the cell and not localized with the compartment. If it is needed for the compartment, we obviously would expect it to co-localize with the compartment. So that's what we expect to see. When we started doing our microscopy things, very important is always to have a control so that you know that what you're doing is actually working. This took us some while to get these images. So when we got them, I was very excited, one could say. And I still think that they're super beautiful, but maybe that's just grandma me coming out. So what this is, GP105 is a protein that was shown in other papers before. And it is a protein that they say is localized in this compartment on the shell. So really the protein structure that builds up the compartment. So we could really show here that it colocalizes with the DNA and with kind of the blob. And also you could really even see the ring structure of it. So that was a very pretty high resolution image that I did not think that we would get to that detail, but that was cool. Anyway, so we got our control, we saw that what we had was working, so we continued with the other 10 genes that we selected for in the very beginning. And from these 10 genes, we actually found 2 others that also colocalized with the compartment or with the DNA. And of course, that needs to be characterized further, but that was really cool because we can infer from that that they're also involved with building the compartment. And that is something that is not published in any other paper yet. So that's actually novel findings that we were able to produce during a student project, which I think is super exciting because I did not think that one could do that during the bachelor's. So as a conclusion, in our lab time, we managed to reproduce findings from a paper, and we found 2 more proteins which seem like they could do something with the compartment. If we were to continue working with this project, which we didn't, one would have to characterize these proteins further, by looking if they're essential for that compartment or if they're even necessary. One could do that with like co-expressing the proteins without infecting and seeing if they form the compartment by themselves already, or by doing knockout phages where you don't have that protein and see if the compartment still forms. But that would be some lab things that one could do. So iGEM is not only about lab though. It's a very exciting part of it and probably my favorite part of it, but it's also about seeing how your project implements to the world and building a community. So we really focused on that part of it. So we really wanted to manage to get a community here in Switzerland that cares about synthetic biology and that promotes iGEM as well. So in the very beginning of our project, so in May, I think, we founded Symbio, which was an important step towards promoting that every year we will be able to have an iGEM team again. And we also did organize the Swiss iGEM meetup with 2 other Swiss teams. So ETH Zurich also had a team, and EPFL. And EPFL actually won the grand prize last year, so that was cool, some Swiss proudness. We also did some collaborations with other iGEM teams to check that our experimental setup was actually working. We talked to some experts about compartmentalization. There is a group at ETH Zurich which works with protein cages, so they were able to give us some input, so the Hilverts team. And then one big part of iGEM is, of course, also the giant jamboree at the very end. So in October, all the iGEM teams meet up. They present their project to the other people in front of judges, which then critically ask you questions and make you panic. You create a poster. You have to present that poster to other students and to the judges who, again, make you panic. And you have to be able to pitch your project that you could talk for hours about, if you ask my friends. And you have to pitch it in 1 minute. So that's also great learning experiences. And so we went to Boston, as Alexander mentioned, which was quite fun and a good learning experience. And in Boston also, in the end, your research kind of gets graded into different levels. And we managed that with our first participation, with being the first team, we managed to get silver level, which is quite exciting, actually. So there's bronze, silver, and gold. And it's not a competitive medal. It's more like you're in this level with all these other teams. You manage to do qualitatively good research, and you manage to have some outreach to the world. But it was a nice thing for a new team because many first teams actually start with bronze. All of this would not have been possible without finding sponsors, which we luckily managed to do. And it also would not have been possible without our supervisors. So we were hosted by the Peltmans Lab. You might know him from the synthetic biology lecture. And we were supervised by Arpon and Adrian. And I think I can confidently say that without the supervision of these two, none of this would have happened. So we're very thankful. So yeah. So that was the part about our project. Here, I want to be honest with you. iGEM projects usually solve real-world problems with synthetic biology. And as you've seen, ours was more a fundamental biology problem with some cool applications sometime later on down the road. That is not the typical iGEM project. And that was partially due to us being the first team and not knowing anything about iGEM before Alexander came to the lecture and told us about it. So yes, what we first had to do is not only do the lab work, get a cool project, but also figure out the iGEM know-how and invest into the team building and into the community building. So that was a big focus point of ours, with the hope of afterwards having the new iGEM teams being better adapted and in a better position for a really cool iGEM project. And I think, at least from what I've heard from the new iGEM team, their project is amazing, and I think they're better adapted to iGEM than we were. So I'm very proud of them. So I also focus more on the project part. If you have any questions about like the personal experience, I will stick around here. So after the presentations are done, all of the involved people will stick around, so you can come up to us, ask us any questions you want, ask us for experiences, ask us for insights, and I will be very happy to talk to you about that.